Hi guys, welcome back to my channel. Thank you for tuning in for another exciting video today. In some of the previous videos, I have spoken about what are motifs, what are transcription factor binding sites. I've also walked through a couple of demonstrations in finding motifs and enriched transcription factor binding sites in epigenomic dataset. And continuing that theme of discussion, we are taking it a little bit ahead in this video and we will be performing an integrated promoter and pathway analysis and identify master regulators using multi-omics data. So this time we will be using GenomeExplain's automated pipeline called Genome Enhancer for performing this analysis. Genome Enhancer is a fully automated pipeline with a point and click interface that identifies transcription factor binding sites, master regulators, prospective drug targets, and corresponding treatments by reconstructing the molecular mechanism of the studied pathology. So, this stands out as the only automated pipeline so far that creates comprehensive insights from start to finish. A big thank you to GenomeExplain for sponsoring this video series as well as giving me the opportunity to explore their platform and automated pipelines. Today we will be exploring their automated genome enhancer pipeline using epigenomics dataset and transcriptomics dataset. If you haven't watched the previous videos in this series, I will add the link to those in the description section below. In order to identify the molecular mechanisms of gene regulation, we should be able to identify binding sites of transcription factor, which are important for all those biological processes. If you have been following me in this series of motif discovery and transcription factor binding site analysis, we went through different ways to identify motifs and transcription factors that bind to those motifs. The next obvious question is to address what to do next after identifying the transcription factor and motifs in our regions of interest. So I want to paint a big picture here for you. The big picture here is the signal transduction network, and let's try to understand where do these transcription factors fit into it. The key component of the regulatory network of the cell is regulation of the gene expression by transcription factors. Because of the central dogma, transcription factors are involved in a feedback loop. DNA is transcribed into RNA, which is translated into protein sequence of a transcription factor, which then binds to the upstream region of the gene and changes its rate of transcription. Transcription factors are vital for the cell's response to its environment because extracellular stimuli can activate a transcription factor via a system of signaling molecules that convey the signal through the relay molecules to the transcription factor. Only when the transcription factor is activated, it will regulate its target gene. A cell receives a signal basically which triggers a response in which this signal is transduced into the cell, resulting in the transcription of the gene. And this is a very simplistic overview of the signal transduction network. In real case scenarios, things are more complex. This figure essentially shows the signaling pathway and transcription factors involved in the HPR1 gene expression in cancer. The mutations or overexpression of the growth factor receptor genes such as EGFR and its downstream signaling molecules such as RAS and BRAF genes result in increased expression and translocation of the ETS family transcription factors such as GAPB and ETS1. And these transcription factors, in cooperation with several other transcription factors that are overexpressed in cancer like SP1, EGR1, lead to HPR expression in cancer, HPR1 expression in cancer. And it becomes clear that gene regulation is accomplished by specific combination of transcription factors rather than by a single transcription factor alone. So that brings us to the idea of composite molecules and master regulators. Master regulators are key proteins or transcription factors that hold significant control over gene expression in biological system. They regulate the activity of multiple genes, often playing a central role in determining cell fate or function. And these regulators can exert their influence by activating or repressing specific genes, thereby orchestrating complex networks of gene expression. Composite modules, on the other hand, refer to the group of genetic elements that work together as a functional unit to control a group of target genes to achieve a particular biological function or response. So these modules often involve interaction between multiple regulatory elements such as transcription factor binding sites within enhancers and promoters that collectively control the expression of target genes of the module. So these genes can exhibit coordinated behavior, responding to various signals or environmental cues to regulate specific cellular processes. So it is helpful to understand and identify these regulators, as identifying composite modules help unravel how group of genes work together to perform specific function or respond to stimuli within cells. Recognizing master regulators associated with diseases can shed light into the molecular basis of illness, aiding in the development of the targeted therapies. Pinpointing master regulators or composite modules associated with certain diseases can lead to discovery of biomarkers for early detection or prognosis. And also, knowing master regulators enables the design of drugs that specifically target these key molecules, which allows us to be more precise and effective in our treatment strategies. In addition to that, uncovering these regulatory elements contributes to the broader understanding of the complex biological systems, which helps in construction of comprehensive treatment models, and also identifying master regulators associated with individual variations help in tailoring treatments to specific patient profiles, enhancing therapeutic outcomes. Lastly, understanding these regulators can prevent individual responses to medications, which helps in selecting the most effective treatment for patients. Genome Enhancer uses upstream analysis, an integrated promoter and pathway analysis to identify potential drug targets of the studied pathology. In the first step of this analysis, the transcription factors that regulate differentially expressed or mutated genes are identified with the use of transfact database of transcription factor binding sites. The second step searches for the most common master regulators of the identified transcription factors by building a personalized signal transduction network of the studied pathology using transpath database of mammalian signal transduction and metabolic pathway. The identified master regulators are prospective drug target candidates. They are used for further selection of chemical compounds that can bring therapeutic benefit for the studied clinical case. So in this step, the human PSD database is employed to identify drugs that have been tested in clinical trials. The cheminformatic tool PASS predicts small molecules that affect the identified targets. Finally, Genome Enhancer generates a comprehensive report about the personalized drug targets that are identified for a certain patient or a group of patients, and the drugs that may be effective in this case. So we will be, for today's video, we will be performing the upstream analysis using the Genome Enhancer, using the epigenomics and the transcriptomic data. And we will be going over the comprehensive insight that will be generated by Genome Enhancer. So for today's demonstration, we will be using the same epigenomic dataset that we used in the previous videos. If you haven't watched them, again, I will be adding the link to those videos in the description section below. The ChIP-Seq data captures the genomic regions where RUNX1 binds, and RUNX1 is a transcription factor that plays a critical role in development of breast cancer. Overexpression of RUNX1 can lead to the activation of genes that promote cancer growth and survival. In terms of the transcriptomics dataset, it's essentially an RNA sequencing dataset which was performed to identify transcripts that are transcriptionally regulated by RUNX1 in RUNX1 wild type and knockout MCF10A cells. The goal of today's analysis is to perform upstream analysis in GenomeExplain's Genome Enhancer to garner insights on the pathways, composite modules, master regulators, and potential therapeutic targets in MCF10A cells regulated by RUNX1. MCF10A cells are non-malignant epithelial cells. This is the ChIP-Seq dataset that we will be using today for the demonstration. This is the same peak set that we perform transcription factor binding site analysis with. So we will be downloading this peak set again for today's demonstration. And for the transcriptomic dataset, this is the transcriptomic dataset that we will be using. And among the files that they provided, we will be downloading this matrix, essentially, which contains unnormalized raw counts from wild type and knockout RUNX1 MCF10A cells. And there are also replicates present within this matrix. I have already downloaded this data, so I'm not going to download it again, but just wanted to show you that these are the files that we will be providing and uploading onto the Genome Enhancer platform and perform further analysis with. So now that we have downloaded our data, let us go onto the Genome Enhancer platform and upload our data and start processing it. So when you log into GenomeExplain, you get access, you essentially see various options to explore their platforms, their databases, and Genome Enhancer tool. So I'm going to click on the Genome Enhancer here. And this is essentially how the Genome Enhancer would look like once you open it. And here you can either start a new project or you can open up an existing project and upload your data and start the analysis. You also have access to demo projects here. So since I have a project under my name already created, I'm just going to open this project. And once you open the project here, you will be able to upload your files for analysis. There are also various acceptable formats. So basically, there are different kinds of multi-omics data that you can upload here. It accepts epigenomic data, transcriptomics, genomics, metabolomics, proteomics, and there are various kinds of formats that it accepts. So here is a detailed list of all of that. In addition to that, they've also provided templates for various kinds of data. So basically, you can download these templates and get your data in the same format and upload. So you would not basically face problems in the tool throwing errors or any discrepancy in the formatting of the data. You can also upload your data from the URL, but since I have files downloaded locally, I'm going to click on the add files and upload my data here. So these are the two files that I said previously that we will be using for today's demonstration. So I'm going to open and upload these files. So each of these files I'm going to import. So the first file is the ChIP-Seq file. Basically, it's the epigenomic dataset. So here we will set the data type to be epigenomics. It's a tabulated file, and based on the previous processing, we have identified that the header row is 30. I can quickly show you how to determine that. So when you look at your ChIP-Seq file here, there are essentially these rows that I want the tool to ignore. So I basically want what line is this row, basically, which is the header row. So there's a very quick way to find that out. So basically the header row starts at row 30. So that's how I know my header row is at row 30. We have done this previously in one of our videos, but for anyone who hasn't watched that and is watching this the first time, then this is a quick way to look up what line is your header row. So the header row is 30. The first row, data row index is 31. The ID, I want it to generate a unique ID, and the rest of it, I'm just going to leave it as is, and I'm going to import. Now let us also import the transcriptomic data. So it detected that it's a tabular format automatically. I'm just going to set it to transcriptomics here. The header row in this case is the first row itself. So we can quickly check that. Since this is a Excel file, we cannot open it here. So let me quickly open it in my system here. So essentially the first is the header row. There are no additional rows over it. So just want to make sure. So first row is the header row. This is the second, the data row starts from the second row. And I also wanted to create a unique ID, and I'm just going to import. So the data has been imported, and we have set certain parameters to import it into the right format. And now that it has been uploaded and imported, we click on the next button. And in the next step, we would essentially want to drag all the files that we want to proceed and further analyze using Genome Enhancer pipeline. But before we do that, here we have the peak set. And so basically this is a tabular format of the peak file that we uploaded. And what we want to do is we need to convert these peaks from the tabular format into a track format. And once we have it in the track format, we will be able to drag them here and use it for further analysis. So essentially, we have done that previously in one of our videos using certain tools available on the GenomeExplain's platform. So before we proceed with the analysis, we would want to get all our data into the right format, and then we would move ahead with the next steps. So let us switch the platform from the Genome Enhancer to the platform here. And now we go to the analysis tab. We go under the methods. We go under data manipulation. And here we will be able to find a utility which will be able to convert the table to track. So essentially, this is the utility that we will want to use to convert the peak table into a track format. So let us go back to the data tab, and here we drag our input table here, and we set the appropriate parameters here. So the from column is the start, the end column is the to column is the end, and we change the sequence collection here. So we go under database, we go under human ensemble, under sequences, we drag the sequence collection here, and the rest of it we just keep it as is. You can rename the track if you want, but I'm just going to go with the default here, and we set this to run. So now that our peak file table has been converted into the track format, we can switch over to the Genome Enhancer now. And in this step, essentially, we perform editing of the data description. So basically, we set a study design here, where basically we tell the Genome Enhancer which samples or which replicates are knockout and which are the wild type. And we set up the study design for further downstream analysis. So I'll quickly show you how we do that. So here we are ready to drop all our files. So just want to make sure that we have our files in the right format. So the peak file has to be of the epigenomics and not the transcriptomics. I'm just want to make sure that we have the right data and in the right format. So the first file that we will be dropping is the peak file, the track file that we generated. And followed by that, we will be importing or dragging and dropping the transcriptomic data. So this is the transcriptomic data that we have. And now that we have dropped all our files that we need to proceed with the following analysis, we click on the next button. So now let us segregate our data into experiment or control, or we can rename this into the knockout and the wild type, depending on the design of the experiment that you have. So here I have the experiment would be runx1 knockout. So I'm just going to rename the element, and here I'm going to rename the element as runx1 wild type. So now I will start segregating my data into these categories. So the ChIP-Seq peak track is essentially the genomic regions in capturing the runx1 binding in MCF10A cells. So this would be my wild type. So for my transcriptomics data, I know there are certain samples that are wild type and there are some that are knockouts. I'm just going to segregate them. So I know that these are the wild type. Now, once I've set up my design experiment and I have segregated my samples into knockout and wild type into two categories, I'm going to click on next. And here I'm just going to set certain parameters. So here I'm going to choose breast neoplasms as the diseases. For the condition, there are two conditions. There is a knockout and there is a wild type. So the condition one is going to be the wild type, and that is going to be my baseline. So I have checked the radio button here next to it. And the second condition is the knockout. And once I have set these parameters, you can add more conditions if you have, but since I have just two conditions and I want to make comparisons between them, I'm just going to keep it to two conditions and set the one, the wild type one as the baseline. And now I'm going to click on start analysis. So this can take a while to run. I have already run this analysis in a different project and I have the report ready. So I'm going to show you the report and we are going to walk through the report and go over each of these sections in further detail. So this brings me to the end of the part 1 of this video. Please stay tuned for the next part where we will be continuing this discussion. If you haven't watched other videos in the series, please make sure you do. I will be adding the link to the previous videos in this series in the description section below.